The third part of this lecture, which we will talk on the humoral immunity or antibody-mediated immunity. So the main character of humoral immunity is played by the B cells. So B coming from the name, from the word, the letter B for B cells coming from bone marrow, which where they will they are produced and also become immunocompetent. So once the B cells become immunocompetent in the bone marrow, they will migrate. Majority of them will migrate to specific sites known as the germinal center, including the white pulp of the spleen and the cortex of the lymph node. It is estimated that 10 million different clones or types of B cells available, each of which can make a different antibody. So we can imagine that the B cells have 10 million different types, which translated that they will be able to recognize at least 10 million types of pathogen or antigen. Humoral immunity involves the activation of B cells into plasma cells. Plasma cells is the one that produce antibodies, and the antibodies then will be released to the blood and lymphatic fluid. So begin with how the HI or humoral immunity activated. So first start with antigen processing and presentation, which to activate the B cells. So activated B cells then will produce antibodies. So we begin with the first mechanism. B cells themselves able to recognize antigen directly. So B cells, each of B cells will have antibody receptors on their surfaces. We already mentioned earlier that there are around 10 million types of B cells. That means we have 10 million type of B cells with different antibody. So there will be one that will be antibody that will match the antigen on the microbe or the pathogen. So once the B cell recognize the antigen, they will process them and then present them the processed antigen exactly like the function of APCs to the helper T cells. B cells displaying processed antigen recognized by T cells, which will release co-stimulators in the form of interleukins. Besides working as like an APCs themselves, I mean B cell also can engulf or detect and process, also display the antigen. APCs also working together to doing their work as antigen presenting cells, engulf, process, digest, and also stick to their surfaces to present to adaptive cells. APCs also presented the antigen, the processed antigen, to B cells. So we have here either the B cell themselves go and recognize the pathogen, or they are being introduced by the APCs. The one that being introduced by APCs, the response is much more intense if the antigens are being processed by APCs. Then we start co-stimulation. which APCs and B cells also present antigen to T helper. So B cell together with APC already meet with the pathogen, process them, and they both will go and present them to T helper cells. This stimulates the T helper to proliferate and secrete cytokines that co-stimulate, mean will stimulate everything including themselves, also the antigen bound B cells. T helper bind to the antigen class 2 complex, then secrete interleukin 2 that acts as a co-stimulator to initiate B cells division and differentiation. Interleukin 2 also acts as an autocrine stimulating proliferation of the same T helper that producing it. So here we can observe there are like three things that can activate B cell. First, they themselves will eat and or stick the antigen to themselves, look for the antigen themselves and process it, and they become activated, or the antigen presented to them by the APCs, or both of these will go and meet T helper, present the antigen to T helper, and then T helper will produce a lot of cytokines to stimulate everything. Here we can observe that APC showing the antigen to the B cells, and they will become activated and proliferate. Also APCs showing the antigen to the T helper, and T helper will produce interleukins to stimulate the B cells to proliferate and produce antibodies. This ability of B cell to recognize and capture antigen by themselves is because they have the antibody receptor. So this antibody receptor can bind to free antigen. Antigen recognition refers to the binding of antigen to B cell antibody receptors. At the clone stage, antibodies do not leave the B cell. So earlier, the inactive form, the antibody will stay at the B cells. The antibodies are embedded in the plasma membrane of the cell, so they are called antibody receptors. B cells, they are monospecific. That means each antibody receptors, each of these antibodies, earlier we already mentioned that B cells have 10 million types, so we can imagine we have 10 million database. So each of the cell clones in the 10 million B cells is specific to one type of antigen, so they are monospecific. So these antibodies on this clone of B cells only specific to one type of pathogen. Each specific type of antigen binds only to those B cells that are programmed to secrete antibodies that attack to the same type of antigen. It become activated when the antibody receptors recognize an antigen. So once activated, the B cells will proliferate and differentiate into plasma cells and also memory B cells. So plasma cells is a type of activated B cells, multiply and cloning itself, turn to plasma cells, which then produce lots of antibodies. It is estimated around 1000 per second the number of antibodies can be produced by one cell. The number of plasma cells goes down after a few weeks once the pathogen is eliminated from the body. Antibodies stay in the blood longer, but eventually the numbers will go down too. As memory B cells, some of the activated B cells will remain in the body as memory cell. Memory cells will divide rapidly as soon as the antigen reintroduced in the future. The pathogen infects again is destroyed before any symptoms shows.